Welcome to the deep dive. It's really quite something how a tiny change in our genetic blueprint, maybe something subtle, can have these massive impacts, way beyond what you normally think of as just a mutation.
Absolutely. It's a whole different scale we're talking about today.
Okay, let's unpack this. Today, we're diving deep into chromosome mutations, variation in number and arrangement. We're pulling insights straight from Essentials of Genetics, the 10th edition.
And like you said, this isn't about those small tweaks, the single-letter typos in the DNA sequence.
No,
Yeah,
exactly. We're talking big picture here. Changes affecting, whole chromosomes or significant chunks of them, adding, losing, rearranging.
precisely. If a gene mutation is like that typo in one word, chromosome mutations are more like shuffling entire paragraphs, deleting chapters, or maybe even duplicating whole sections of the book.
Wow, okay. That's a big difference.
And the implications are, well, profound. So our mission today is to really get into these chromosomal aberrations. How do they happen? What do they mean for an organism?
And how do they play into bigger things like evolution and human health?
Exactly. We'll clarify the key terms and try to connect these sometimes complex ideas to things we see in the real world.
Great. So let's start with the basics. What exactly are these chromosome mutations or chromosome aberrations? How are they distinct?
Well, the core distinction, as we mentioned, is scale. They involve changes in the total number of chromosomes an organism has.
Okay, like having too many or too few.
Right. Or they involve major structural changes, large deletions, getting extra copies of big segments, or pieces of chromosomes ending up in totally the wrong place.
It really highlights how balanced everything needs to be, doesn't it? This genetic equilibrium.
It really does. Even seemingly minor shifts at this level can cause noticeable phenotypic variations and substantial changes. They're often lethal, particularly in animals.
Okay, so let's talk about those number variations. You mentioned two main types.
That's right. First up is aneuploidy. That's where an organism gains or loses one or more chromosomes, but crucially, not a complete set.
So if the normal is, say, 2N for a diploid organism.
Aneuploidy would be something like 2N plus 1 chromosome or 2N minus 1, that kind of thing.
Right, and there are specific terms for those.
Yes. Losing a single chromosome is monosomy, that's 2N minus 1.
Okay.
And gaining a single chromosome is trisomy 2N plus 1. You can also have the trisomy 2N plus 2 and so on, but monosomy and trisomy are the most discussed.
Got it. And the second category?
That's euploidy. This term describes organisms that have complete haploid sets of chromosomes. So N is one set.
Like us. We're diploid, 2N, two set.
Exactly. But sometimes organisms can have more than two complete sets. That's polyploidy.
So like 3N, 4N, 5N, multiple full sets.
Precisely. Triploidy, tetraploidy, and so forth.
So how do these changes in number actually happen? You mentioned it starts with a slip-up during cell division.
Yeah, it typically stems from an error called nondisjunction.
Nondisjunction.
Okay, basically the failure of chromosomes to separate properly during meiosis. That's the cell division process that makes sperm and eggs.
Ah, okay, so it's an error in making the reproductive cells.
Exactly. Normally, the paired homologous chromosomes separate, so each gamete gets one copy, or later, sister chromatids separate. But if non-disjunction happens,
They stick together.
right, one gamete might end up with an extra chromosome, N plus 1, and another might end up with none for that chromosome, N1.
And then if that gamete is involved in fertilization,
You get a zygote, the first cell of the offspring that's either monosomic 2N1 or trisomic 2N plus 1 for that specific chromosome. It sets the stage right from the beginning.
okay, let's dig into aneuploidy more. What about monochemy losing that one chromosome? You said it's usually bad news.
Very much so, especially in animals like us. The best-known human example that survives is Turner syndrome, where individuals have just one X chromosome, 45X.
But for the other chromosomes, the autosomes.
Autosomal monosomy in humans is generally lethal. It's just not tolerated. Even in fruit flies, monosomy for the larger chromosomes is lethal.
Why is losing one chromosome so much worse often than gaining one?
There are a couple of key reasons. First, think about recessive lethal alleles. If the one copy of the chromosome you have carries a lethal recessive gene,
There's no second normal copy to mask it.
exactly, it gets unmasked, and that can be fatal. The second reason is haploinsufficiency.
Haploinsufficiency.
Yeah, it means that for some genes, having just a single copy simply doesn't produce enough of the necessary protein product for the cell or organism to function properly. One copy isn't sufficient.
Right, like needing two workers for a job and only having one show up.
That's a decent analogy. Interestingly, plants tend to tolerate aneuploidy, including monosomy, A bit better than animals. You see it in maize and tobacco, though they're often less viable, less healthy.
Interesting difference. What about trisomy then, gaining an extra chromosome?
Generally, trisomy 2n plus 1 is tolerated a bit better than monosomy, especially if the extra chromosome involved is relatively small.
But still potentially problematic.
Oh, definitely. In animals, having an extra copy of a large autosome is usually lethal too. But again, plants show more flexibility. The Junson weed, Datura, is a classic example where having an extra copy of different chromosomes leads to distinctly different seed pod shapes.
Okay, and this brings us to a really important human example, Down syndrome. That's a trisomy, right?
Yes, Down syndrome is trisomy 21. It's the most common human autosomal trisomy that survives, occurring in about 1 out of every 800 live births.
First described way back in 1866, I believe.
By John Langdon Down, yes. Individuals typically have a recognizable set of characteristics, though usually only a subset of the potential 12-14 features. Things like the epicanthic fold by the eyes, a flatter facial profile, short stature, and varying levels of intellectual and developmental delays.
And there are associated health issues, too.
Yes, often. A higher risk for respiratory problems, heart malformations are common, and a significantly increased risk, about 20 times higher, of leukemia. Also, early onset Alzheimer's disease is much more prevalent.
But you mentioned something surprising earlier, a protective effect.
Right, the flip side. Individuals with Down syndrome actually have a lower risk of developing many types of solid tumors, like lung cancer or melanoma.
How does that work?
It seems linked to an extra copy of a gene called DSCR1 on chromosome 21. This gene helps regulate, and in this case suppress, angiogenesis, the formation of new blood vessels.
So it makes it harder for tumors to get the blood supply they need to grow.
That seems to be the mechanism, yes. It's a fascinating example of the complex effects of gene dosage.
And where does this extra chromosome 21 usually come from?
Overwhelmingly, it's due to nondisjunction during meiosis. About 75% of the time, the error happens in meiosis site. And interestingly, about 95% of cases, the extra chromosome comes from the mother's egg, not the father's sperm.
Which connects to the well-known link with maternal age, right?
Exactly. The incidence of Down syndrome rises sharply as maternal age increases. It's about 1 in 1,000 for mothers at age 30, jumps to 1 in 100 by age 40, and maybe 1 in 30 by age 45.
Why is that? What's the hypothesis?
The leading idea relates to the long time human eggs are arrested in meiosis I. In older mothers, the eggs have been suspended in this state for decades, and perhaps the cellular machinery for chromosome segregation becomes less efficient or more prone to errors over time.
So this really gets to the core question. What is it about just one extra chromosome, especially a relatively small one like 21, that causes such a significant developmental impact?
This is the central puzzle. Research points to a specific area on chromosome 21 called the Down syndrome critical region, DSCR. The hypothesis is that having an extra dose of certain genes within this region is responsible for many of the syndrome's characteristics. It's about gene dosage sensitivity.
And we now have ways to detect this before birth.
Yes, prenatal diagnosis options include amniocentesis and chorionic villus sampling, CVS, which analyze fetal cells. More recently, there's non-invasive prenatal genetic diagnosis, or NIPGD, which analyzes fetal DNA fragments found in the mother's blood.
Are there other human trisomies that allow survival to term?
Only 2 others, really. Patau syndrome, which is trisomy 13, and Edwards syndrome, trisomy 18. Both involve severe malformations and, sadly, most affected infants don't live long past birth. It underscores how disruptive most aneuploidies are. About 30% of all spontaneous abortions involve some form of chromosome number abnormality.
Okay, let's pivot now to the other category. Polyploidy. Having more than 2 complete sets of chromosomes.
Right, so we're talking 3N, 4N, etc., triploid, tetraploid.
And you said this is much more common in plants.
Far more common in plants. It does occur in some animals, certain lizards, amphibians, fish, but it's really a major feature in plant evolution and agriculture.
Does the number of sets matter? Like, is 3N different from 4N in terms of stability?
Yes, significantly. Odd numbers of sets, like triploids, 3N, tend to be unstable reproductively. It's very difficult for them to produce balanced gametes through meiosis, where each gamete gets exactly 1.5 sets, so they're often sterile.
Which is why things like seedless watermelons or bananas are often triploid. They can't make viable seeds.
Exactly. That sterility is often commercially desirable. Even numbered polyploids, like tetraploids, 4N, can often be fertile because they can potentially produce balanced 2N gametes.
So polyploidy seems like hitting copy-paste for the entire genome, and plants just handle it better. How does it arise?
There are two main pathways. The first is autopolyploidy.
Auto, meaning self.
Yes, the additional chromosome sets are identical to the normal sets of that species. It might happen if, say, all chromosomes fail to segregate during meiosis, resulting in a diploid 2N gamete instead of a haploid N1. If that fuses with a normal haploid gamete, you get a triploid 3N zygote.
Or maybe two sperm fertilizing one egg.
That's another possibility. We can also induce it artificially. A chemical called colchicine is often used.
How does colchicine work?
It disrupts spindle fiber formation during mitosis. So the chromosomes replicate, but the cell can't divide. If it then re-enters the cell cycle, it has double the original chromosome number.
And what's the effect of having all these extra sets?
Often, autopolyploids have larger cells, which can translate to larger overall size, bigger flowers, bigger fruits. Many important crops are polyploids, like potatoes, winesap apples, alfalfa, coffee, peanuts. Even those big strawberries are often octoploid.
Wow. Okay.
And the second pathway,
Is allopolyploidy, also sometimes called amphitoploidy.
allo, meaning different.
Right. This involves combining chromosome sets from different, though usually closely related species, typically through hybridization.
So you cross species A with species B.
Exactly. Let's say species A has the sets AA and species B has BB. Their hybrid offspring would have chromosome sets AB. Now, these chromosomes aren't homologous. They can't pair up properly in meiosis, so this initial hybrid is usually sterile.
Okay, makes sense.
But if that sterile hybrid then undergoes a spontaneous or induced chromosome doubling event,
Like with colchicine again?
could be, then you get an AABB organism. Now, every chromosome does have a homologous partner, an A with an A, a B with a B. This organism, the amphitoploid, is often fertile.
Can you give an example? A classic one is cultivated American cotton. It's an allopolyploid that arose from a natural cross between an Old World cotton species with 13 large chromosome pairs and a wild American species with 13 small pairs. The initial hybrid was sterile, but after chromosome doubling, it became the fertile 26-pair species we grow today.
Fascinating. It's like creating a new species by merging genomes.
It essentially is. Another great example is Triticale. It's a human-made allopolyploid, a hybrid between wheat, tetraploid, and rye, diploid. It combines desirable traits from both wheat's yield and protein, rye's hardiness, and lysine content.
So humans have really harnessed polyploidy.
Absolutely. For agriculture, horticulture, it's been incredibly important.
Okay, so we've covered changes in chromosome number. What about changes to the structure of individual chromosomes?
Right, the second major class of aberrations. These involve changes that delete, add, or rearrange substantial portions within or between chromosomes.
How do these happen?
They usually start with one or more breaks along the chromosome. Chromosome ends that are broken are sort of sticky, they can rejoin, but sometimes they rejoin incorrectly, or a piece gets lost entirely.
And what happens if an individual inherits one normal chromosome and one with a structural change? They're heterozygous for it.
Exactly. This can lead to issues during meiosis because the chromosomes might not be able to pair up perfectly along their entire length. This can result in gametes that are genetically unbalanced, missing some genes, having extras of others, which often affects the viability of offspring, even if the parent carrier is perfectly healthy because they haven't actually lost or gained any genetic material, just rearranged it.
Let's break down the types. First, deletions.
Simplest one conceptually. A part of a chromosome is just lost or deleted. It could be near the end, terminal, or somewhere in the middle, intercalary.
And consequences.
The piece that's lost, if it doesn't have a centromere, usually just drifts away and degrades.
Hmm.
Even losing a small piece can have significant effects. Larger deletions are often lethal.
Is there a human example?
A well-known one is cri-du-chat syndrome. It's caused by a deletion of a small part of the short arm of chromosome 5.
Cri-du-chat. Cry of the cat.
Yes, infants with this syndrome have a distinctive high-pitched cry that sounds like a cat meowing due to larynx issues.
Yeah.
They also typically have intellectual disability, developmental delays, and characteristic facial features. It's a really stark example of how losing just a small fragment can have such drastic developmental consequences.
Heartbreaking. Okay, what about the opposite duplications?
Duplications are when a segment of a chromosome is present more than once. So you have extra copies of some genes,
How do they arise?
often through errors during meiosis, like unequal crossing over between homologous chromosomes. One chromosome ends up with a deletion, and the other gets a duplication. Or it could be a mistake during DNA replication.
What are the implications of having extra gene copies?
Several important ones. Gene redundancy is key. Sometimes you need multiple copies of essential genes, like those for ribosomal RNA, because the cell needs to make so much RNA.
Right, cells need tons of ribosomes.
Exactly. E. coli has 7 copies of the RNA genes. Drosophila has over 100. But the really profound role of duplications is in evolution.
How so?
There's a famous hypothesis by Susumu Ohno. He proposed that gene duplication is a major driving force for creating new genes with new functions.
Oh, okay, explain that.
Think about it. If a gene performs a vital function, it's hard for it to mutate and acquire a new function without potentially harming the organism, right? Because the original function might be lost.
Sure, it's constrained.
But if that gene gets duplicated, the original copy can continue doing its essential job. The extra copy is then somewhat redundant. It's free to accumulate mutations over evolutionary time.
Ah, so it can experiment, genetically speaking.
Precisely. It can diverge and potentially eventually evolve a new beneficial function. This provides the raw material for evolutionary innovation.
Do we see evidence of this?
Absolutely. We see it in gene families, groups of related genes with similar sequences, like the globins that make hemoglobin and myoglobin. They clearly arose from ancestral duplications. A really striking recent example is the SRGAP2 gene, involved in brain development.
How about it?
Humans have 4 copies of this gene, while other primates only have 1. The timing of these duplication events seems to correlate with key points in human evolution, like the expansion of the brain and the development of language and complex cognition.
Wow, so duplications might be partly responsible for making us human.
It's certainly a compelling piece of the puzzle. And related to this, we now know about copy number variations, or CNVs.
CNVs.
Yeah, with modern sequencing, we've discovered that duplications and deletions of segments ranging from small to quite large are actually incredibly common in genomes. The number of copies of certain genes or regions varies between individuals.
So we don't all have exactly 2 copies of every gene region.
Not at all. These CNVs are widespread and are now understood to be really important. They contribute to normal human variation, but they're also linked to susceptibility to various diseases, autism, neurological disorders, cancer, even things like Crohn's disease risk or HID progression.
Fascinating. So duplications are much more dynamic and important than maybe we first thought.
Definitely. They're not just errors, they're sources of variation and evolutionary potential.
Okay, next structural change. Inversions, what are they?
An inversion is when a segment of a chromosome gets flipped 180 degrees. So the linear sequence of genes in that segment is reversed.
Like taking a sentence, cutting out a phrase, flipping it, and sticking it back in.
Exactly like that. All the genetic information is still there, just rearranged. No loss or gain of material, usually.
How does that happen?
Typically requires 2 breaks in the chromosome. The piece between the breaks gets inverted before the ends are rejoined.
Are there different types?
Yes, based on whether the centromere is included in the inverted segment. If it's not, it's a paracentric inversion. If the centromere is within the flipped part, it's pericentric.
Does this cause problems? The information is still there.
The individual carrying the inversion might be perfectly fine. The problems arise during meiosis, if they are heterozygous for the inversion, 1 normal, 1 inverted chromosome.
Why? How do they pair up?
They have to form a characteristic inversion loop to allow the homologous segments to align as much as possible.
A loop.
Now, if crossing over happens within that loop, things get messy, especially with paracentric inversions.
What happens?
A single crossover inside a paracentric inversion loop produces some really weird chromatids. One ends up with 2 centromeres, dicentric, and another ends up with none, acentric.
2 centromeres. No centromere. That sounds bad.
It is. The dicentric chromatid gets pulled in 2 directions and breaks. The acentric one gets lost. Both resulting gametes are genetically unbalanced, missing genes, having duplicates. They usually lead to non-viable offspring.
So the crossover products essentially get eliminated.
That's right, which is why inversions are sometimes said to suppress crossing over. You don't stop it from happening physically, but you don't recover the recombinant offspring because they're inviable.
Is there any advantage to inversions then?
Evolutionarily, yes. By preventing the recovery of crossover products within the inverted region, an inversion can lock together a specific set of advantageous alleles on that chromosome, preventing them from being broken up by recombination. They can act like supergenes.
Interesting. Okay, last category, translocations.
Translocations involve moving a segment of a chromosome to a completely different location in the genome, often onto a non-homologous chromosome.
So moving a piece from chromosome 3 to chromosome 8, for example.
Exactly. The most common type is a reciprocal translocation, where two non-homologous chromosomes exchange segments.
Is genetic material lost or gained?
Not usually in the reciprocal exchange itself. So,
Okay.
again, the carrier might be phenotypically normal. They have all the right genes, just rearranged.
But problems arise in meiosis again.
Yes. When the chromosomes try to synapse in a translocation heterozygote, they form a characteristic cross-like configuration because they're trying to maximize pairing between homologous regions that are now on different composite chromosomes.
That sounds complicated for the cell to sort out.
It is. Depending on how those chromosomes segregate during meiosis I, you can get different outcomes. Some segregation patterns lead to balanced gametes, either normal or carrying the balanced translocation, but others, called adjacent segregation patterns, lead to unbalanced gametes with duplications and deletions.
And unbalanced gametes mean?
Reduced fertility, often called semi-sterility. About half the offspring might be inviable due to the genetic imbalance. In humans, this can manifest as recurrent miscarriages or the birth of children with abnormalities resulting from partial monosomy or trisomy for the translocated segments.
This feels like it connects back to Down syndrome somehow. You mentioned a familial type.
Exactly. This is distinct from the common trisomy 21 caused by nondisjunction. Familial Down syndrome runs in families and is caused by a specific type of translocation called a Robertsonian translocation.
What's that?
It involves two specific kinds of chromosomes, acrocentric ones, which have the centromere very near one end. The long arms of two different acrocentric chromosomes fuse near the centromeres, and the tiny short arms are lost.
Okay.
The most common one related to Down syndrome involves chromosomes 14 and 21. A parent might carry a 14-21 translocation. They have the fused 14-21 chromosome plus a normal 14 and a normal 21.
So they effectively have only 45 chromosomes because 14 and 21 are joined.
Correct. But because the lost short arms carry very few genes, this carrier is phenotypically normal. They have pretty much the full genetic complement, just packaged differently.
But when they make gametes?
That's where the risk comes in. Due to different segregation possibilities of the fused 14-21 chromosome and the normal 21, they can produce several types of gametes. Some are normal, some carry the balanced translocation, but one type leads to offspring with Down syndrome. These kids will have 46 chromosomes total, but because they get the fused 14-21 from the carrier parent plus a normal 21 from the other parent, they effectively have 3 copies of the critical chromosome 21 material.
Wow, that really shows how complex inheritance can be. A parent with 45 chromosomes can be normal, but have a child with Down syndrome who has 46.
It requires careful genetic analysis to understand these situations.
Okay, let's touch on one more specific phenomenon, fragile sites.
Right. These were discovered in the 1970s. They're specific locations on chromosomes that appear as gaps or constrictions when cells are cultured under certain conditions, like low folic acid.
Yeah.
They seem to be regions where the chromatin isn't packed as tightly.
And they're fragile because
They are susceptible to breakage. Associations have been found between these fragile sites and conditions like intellectual disability and also with cancer.
Is there a main example linked to intellectual disability?
Yes, the most common form of inherited intellectual disability is Fragile X syndrome, FXS. It affects roughly 1 in 4,000 males and 1 in 8,000 females.
What are the characteristics?
Besides intellectual disability, common features include a long, narrow face, prominent jaw, large ears.
And what's the genetic basis? It's at a fragile site on the X chromosome.
Yes, specifically at a site called FRXA. The underlying cause is fascinating. It involves an expansion of a trinucleotide repeat, specifically CGG repeats, within a gene called FMR1.
Trinucleotide repeats, so the sequence CGG repeated over and over.
Exactly. Most people have between 6 and 54 copies of the CGG repeat in the FMR1 gene.
Okay.
Individuals who are carriers, sometimes called premutation carriers, have about 55 to 230 repeats. They usually don't have FXS themselves, but the repeats are unstable and can expand further when passed on to offspring, especially when passed by the mother.
And if it expands beyond that?
More than 230 repeats generally leads to the full Fragile X syndrome. This increase in repeat number across generations is called genetic anticipation. The condition tends to become more severe or appear earlier in successive generations.
How do the extra repeats cause the syndrome?
The excessive number of CGG repeats triggers a process called methylation in that region of the gene. This effectively silences or inactivates the FMR1 gene.
So the gene is there, but it's turned off.
Right. The FMR1 gene normally produces a protein called FMRP, which is important for normal brain development and function. In FXS, the lack of this protein leads to the observed cognitive deficits and other features.
And you mentioned a link between fragile sites and cancer, too?
Yes, that connection became clear in the mid-90s. A fragile site on chromosome 3 called FRA3b contains a gene called FHIT. This gene is often found to be deleted or altered in cells from many types of cancers: lung, breast, cervical, colon cancer, and others.
So the fragility makes the genes within those regions more vulnerable.
It seems so. Genes located in fragile sites appear more susceptible to being mutated or deleted, and loss or damage to these genes can contribute to the development of cancer. It's another way chromosome instability impacts health.
This all brings us to some really heavy territory, doesn't it? Especially when we think about detection and choices.
It absolutely does. Our increased understanding of these chromosomal abnormalities, especially through prenatal testing for conditions like Down syndrome, forces us to confront some profound ethical questions.
Yeah, the availability of prenatal tests, which can lead to decisions about continuing a pregnancy, is a really sensitive area.
Extremely. And there are deeply held, often opposing viewpoints. Some people argue that preventing the birth of a child with a significant genetic condition is a morally acceptable way to reduce potential suffering.
While others argue strongly against it, saying that selective termination based on genetic makeup is unethical, perhaps even bordering on, well, eugenics.
That term, eugenics, carries a lot of historical weight. Coined by Francis Galton back in 1883, it originally referred to improving the inborn qualities of a population, often through selective breeding.
But it get twisted into some horrific practices in the 20th century.
Absolutely. Compulsory sterilization programs in the US, UK, Canada, and most notoriously, the policies of Nazi Germany completely discredited the concept and the term itself.
So the question arises, and our source material prompts us to consider this. Is modern prenatal diagnosis followed by selective termination a form of eugenics? Why or why not?
It's a complex debate with no easy answers. And it extends further. If technology allowed us to select for certain traits in our children, height, intelligence, whatever, would that be ethical? Would that be eugenics?
These are tough questions that society is grappling with as our genetic knowledge grows. We're not here to provide the answers, of course, but to present the dimensions of the discussion raised in the genetics field itself.
Precisely. It highlights the intersection of science, ethics, and societal values.
So wrapping this up, what a journey. We've gone from these tiny slips in meiosis leading to extra or missing chromosomes like in Down syndrome
To large-scale rearrangements like deletions, duplications, inversions, translocations, reshaping chromosomes and impacting health, inheritance, and even evolution itself.
It really makes you appreciate the incredible intricacy and maybe the fragility of our genetic blueprint, how the precise number and structure of these chromosomes is usually so critical.
It is, and yet life also shows remarkable resilience and finds ways to incorporate variation, sometimes using these very changes as fuel for adaptation over long timescales.
So the next time you think about genetics, maybe picture that complex dance of the chromosomes. Consider how a subtle misstep or a rearrangement can rewrite not just one trait, but potentially a whole biological narrative. It's a fine line, isn't it, between variation and vulnerability?
It truly is. Thank you for joining us for this deep dive. We hope exploring these chromosome mutations has given you a clearer picture and perhaps even more curiosity about this amazing field.
We certainly hope it helps you feel well informed. That's it for this deep dive. Until next time, keep digging.